Hi, my name is Neil Singleton and I'm the Insurance Commissioner with the Motor Accident Insurance Commission here in Queensland. Today I'm proud to announce the winners of Bionics Challenge 2021, a unique Queensland-led competition designed to fast-track medical bionic innovations to change the lives of those living with serious injuries or loss of vital functions as a result of road trauma, complex disabilities and/or chronic disease. The 4 winning teams that will each receive $10,000 to further advance their innovation include Central Queensland University's Dr. Vanessa Bokazanian and her team of innovators from Australia, India and South America who are advancing e-stimulation therapies for people impacted by neurological trauma and spinal cord injuries. University of Queensland biomedical innovator Dr. Antonio Bo and his collaborators at UQ and Metro North Hospital and Health Service, who are developing VR/AR-enabled sensory rehabilitation to accelerate occupational therapy for individuals with upper limb conditions. 2 Brisbane-based brothers and designers, Andrew and David Peterson, who are developing Gyrite, a haptic wearable that gives people with severe balance dysfunction the sensory feedback they need to stay upright. Queensland Brain Institute research leader Dr. Matilde Balbi and her team who are developing an individualised treatment for stroke recovery. This integrative and innovative system is set to employ automated monitoring of brain activity and AI-enabled computations to inform a customised approach to brain stimulation for stroke survivors.
Hi, I'm Dr. Matilde Balbi and I'm a group leader at the Queensland Brain Institute working on methods to promote stroke recovery. Stroke is a leading cause of death and disability in Australia and is one of the highest burdens on the health system nationwide. About 15 million people have a stroke annually, and that accounts for roughly 3% of the total global healthcare expenditure. Surprisingly, there has been very little progress in the development of stroke recovery technology since the discovery of a tissue plasminogen activator, which is only effective when delivered to patients within the first 5 hours after stroke. No 2 strokes are identical. The current one-size-fits-all approach results in a large difference in treatment outcomes between patients. Designing more personalized treatment approaches tailored to address the individual's need could lead to improved outcomes for stroke survivors. In this presentation, we will describe our approach to develop protocols for using the individual brain activity of stroke patients to fine-tune a set of stimulation parameters to aid stroke recovery. Optogenetics is a scientific method in which light is used to activate brain cells. We have performed initial experiments in mice where we used optogenetics techniques to stimulate groups of brain cells at different frequencies and found that we were able to aid both motor and neurological recovery following stroke. This approach enables us to identify specific frequencies that can be used as stimulation parameters in future experiments. Due to the regulations surrounding the use of optogenetics in human patients, our plan is to implement non-invasive brain stimulation as an alternative approach. Our experimental team is made up of neuroscientists, Montana Semantics and Lucy Tatum-Heap, who are designing artificial intelligence technologies which will allow for real-time recording of brain activity to shape the parameters of individualized brain stimulation.
Our research is trying to expand on currently used therapies for stroke recovery in patients using brain stimulation. Transcranial alternating current stimulation is a type of non-invasive brain stimulation that adjusts brain activity through electrical currents at a specific frequency. This kind of stimulation has been used experimentally to treat stroke. However, most of these studies use predetermined parameters for their stimulation protocol. This means that they stimulate everybody at the same settings, leading to a large range of variations and a low success rate. To overcome this, we are developing an individualised protocol that modulates and delivers brain stimulation based on individual brain activity. This is our modular automated rehabilitation system. In this, we can monitor patterns of brain activity associated with behaviours in real time using machine learning methods in awake and behaving animals. Mice will remain in the rehabilitation system for up to 3 months after stroke, and both their brain activity and motor functioning will be assessed through the automated system. Within our system, we use a microscope to record brain activity and analyse this in real time using machine learning methods. The system will perform real-time analysis on this brain activity and will deliver brain stimulation when the mouse performs a motor task. The performance of animals and their brain activity will be used alongside artificial intelligence computations, which will fine-tune these stimulation parameters based on the brain activity. Because of this, we rely on an in-lab high-performance computer that will be capable of analysing brain activity online as it is collected, which will be used to modify the brain stimulation parameters in real time as the mouse is performing the task. By measuring the brain activity of each animal, we can tailor our brain stimulation protocol and hopefully improve the chances of a successful treatment. This cutting-edge treatment plan will also be individualised for human patients in clinical trials.
We aim to complete our pilot experiments by 2023, and by the end of 2022, we'll have an initial and compelling data set to inform future human trials. As we are currently starting our preliminary data collection, this timeframe will allow us to collect all required experimental groups with the relevant controls and parameter variations. With the purchase of a second automated rehabilitation system, this would double our throughput of data collection. The team will continue to work closely with UniQuest, the commercialization company of UQ, to manage the IP strategy and licensing to determine how the research findings can be translated to patients. Potential industry partners that work in the field of brain stimulation for stroke recovery include Sorterix, Neurocon, and Neuroelectrics. We expect that they will assist in the design of a brain-computer interface that will both record brain activity and deliver an individualised protocol tailored to the patient. There is a clear need for non-invasive treatment options for stroke patients that can be applied within a short time window. The target market for our end product will be stroke survivors, which within Australia alone could include around 27,000 people per year. Worldwide, this number increases to about 16 million people. Future target markets for our product include patients with related neurodegenerative disorders, including dementia.
Our approach will pave the way to the much-needed use of artificial intelligence-driven systems to reduce the high variability in treatment efficacy currently observed in patients. We will fine-tune a set of stimulus parameters based on the individual brain activity of patients, which we hope will be able to drive recovery following stroke.
So we're now congratulating Dr. Matilde Balbi and her very talented team from the Queensland Brain Institute based at the University of Queensland. And Matilde and her team are working on the development of an individualised closed loop approach for stroke recovery. And I think this is a fascinating project because of that AI-enabled monitoring of brain activity and then looking at the customised approach to therapy. So Matilde, if we can actually ask you and the team, wonderful team assembled there, just some of the things that having won this $10,000 prize that you'll be able to move forward with. It'd be great to hear that.
Yeah, first of all, thanks a lot for giving us the opportunity to be here, and we learned a lot during the entire process, and it has been great. So something that the prize will allow us to do is to move forward with our research and get a high-performing computer that will allow us to really visualize and send feedback online, which is something that it's key for our project. And this will allow us to having our own in our own lab, and that's just fantastic.
No, I really love the fact that we've got a talented group of young female neuroscientists here that are really forging ahead. And Matilde, I think you've got a new laboratory or new group that you've, do you want to tell us a little bit about that?
Yeah, my group is very young. We just started here, and we're going to celebrate 1 year from the opening of the lab here at the Queensland Brain Institute. And yeah, for us, winning this prize is really like, a good starting point that validates the kind of research that we do and the importance of the research that we do. We obviously always look forward to implementing what we do in the clinics, and this is, winning this prize really tells us that we're going in the right direction. And thanks again.
No, that's great. And I think we're looking forward to also showcasing your work there at the Queensland Brain Institute over coming months. So congratulations again.